Why is optic nerve damage difficult to regenerate? Challenges and advances in neuroscience. Optic nerve damage is difficult to regenerate because of several biological, structural, and molecular barriers inherent to the central nervous system, which includes the optic nerve. Limited regenerative capacity of the central nervous system, CNS. Unlike peripheral nerves, CNS neurons, including retinal ganglion cells, or GCS, that form the optic nerve, have a limited ability to regenerate. This is partly because mature CNS neurons lack the intrinsic growth capacity they exhibit during development. Inhibitory environment. After injury, the CNS creates an environment that inhibits nerve regeneration. This includes myelin-associated inhibitors, molecules like NOGO-A, myelin-associated glycoprotein, and oligodendrocyte myelin glycoprotein inhibit axonal growth. Astrocytes and other glial cells form a dense scar at the injury site, producing molecules like chondroitin sulfate proteoglycans that further block axon regrowth. Loss of retinal ganglion cells. Optic nerve damage often leads to the death of retinal ganglion cells, the neurons whose axons make up the optic nerve. Once these cells die, they cannot be replaced, limiting the potential for regeneration. Lack of growth-promoting signals. Developmental growth-promoting factors, like neurotrophic factors, are downregulated in the adult CNS. In addition, injured neurons often fail to express genes associated with axon growth. Immune response. The immune response to injury in the CNS is different from that in the peripheral nervous system. Microglia and macrophages, which are involved in clearing debris, may not function efficiently in the optic nerve, leading to persistent inhibitory molecules. Complex architecture of the optic pathway. The optic nerve connects the retina to the brain through a highly organized structure. Regenerating axons must not only grow over long distances, but also re-establish precise connections with target neurons in the brain. Achieving this level of specificity is extraordinarily difficult. While regeneration is challenging, researchers are exploring ways to overcome these barriers, such as gene therapy, enhancing the expression of growth-associated genes in our GCS. Neurotrophic factors, administering proteins that promote neuron survival and axon growth. Blocking inhibitory molecules, developing drugs or antibodies to neutralize molecules like NOGO-A or CSPGS. Stem cell therapy, using stem cells to replace lost RGCS or create a supportive environment for regeneration. Microcurrent stimulation, promoting neural activity to enhance axon growth. AC STIM involves the application of low-level electrical currents to the optic nerve cells, aiming to stimulate cellular activity and enhance retinal cell function. It also improves blood flow to the retina by promoting vasodilation, the widening of blood vessels. Better blood flow could enhance nutrient and oxygen delivery to the optic nerve cells, supporting their health and potentially slowing degeneration. The stimulation also encourages the release of neuroprotective factors that help preserve retinal cells and slow the progression of optic nerve atrophy.